Equisetum, a pteridophytic genus, commonly known as horsetail or scoring rush, commonly grows in marsh places, ponds, or along the sandy river banks. The genus is distributed all over the world except Australia and New Zealand. The genus is represented world over by 30 species. In India, there occur 8 species. Of these, Equisetum arvense, Equisetum debile, Equisetum diffusum, and Equisetum elongatum are the most common and widely distributed one. Species of Equisetum are used as indicators for minerals in the soil. Some species accumulate gold from the soil. There is a deposition of silica on the outer walls of epidermal cells, due to which the plants become rough in texture and are used as abrasive. Silica also provides a protective covering to these plants against predators and pathogens. Some species of Equisetum are also used in Ayurvedic medicines as diuretic. Equisetum belong to kingdom Plantae, division Pteridophyta, subdivision Sphenopsida, class Lycophyta, order Equistales, family Equistaceae, and the genus Equisetum. The sporophyte of Equisetum has a perennial, branched and creeping underground rhizome, which gives annual erect aerial branches. The sporophyte of Equisetum is differentiated into stem, root, and leaves. Let us discuss first its stem, which is also called as rhizome. The main stem is underground, creeping and perennial rhizome. It often lies more than a meter below the surface of the soil. The stem is differentiated into nodes and internodes. There is a whorl of small scaly leaves at each node. These leaves are joined at their bases, forming sheath around the node. The rhizome bears 2 types of aerial shoots: vegetative shoots and fertile shoots. Both these types of shoots are differentiated into nodes and internodes. The vegetative shoots are usually branched, green, and photosynthetic, while as the fertile shoots are unbranched and achlorophyllous, each terminating into a cone or strobilus. The vegetative branches are of following 2 types. First, branches of unlimited growth. They are further branched like the main vegetative shoot and are differentiated into nodes and internodes. Branches of limited growth. They are also differentiated into nodes and internodes like vegetative shoots, but remain unbranched. The leaves of Equisetum are minute, scaly, and isophilous. They arise in whorls at the nodes of both the underground rhizome and aerial branch. These leaves are fused at their bases to form a distinct sheet, and their distal free ends give a frill-like appearance. The leaves of Equisetum are non-photosynthetic, and their main function is to protect the branch bud at their node. The anatomy of the stem of Equisetum is interesting, as it shows a combination of hydrophytic as well as xerophytic characters. The internodes and nodes have different internal structures. Let us first discuss the internal structure of internode. When we cut the transverse section of internode, it is wavy in outline due to presence of ridges and grooves. It has a distinct epidermis, a well-developed cortex, a stele with a ring of vascular bundles, and a large central pith cavity. The epidermis is composed of a single layer of cells with a heavy incrustation of silica on their outer and radial walls. Silica also provides mechanical strength to the shoot. The epidermis is interrupted by stomata, which are usually confined to the grooves in case of Equisetum. The cortex is differentiated into an outer and an inner region. The outer cortex consists of sclerenchymatous and chlorenchymatous cells. The sclerenchyma lies just below the epidermis and provides mechanical strength to the shoot. It is many layered below the ridges, but in the region of grooves, it is only 1 to 2 layered. The inner cortex is composed of thin-walled parenchymatous cells. It has a large schizolysogenous canals known as vallicular canals below the ridges. These canals extend throughout the length of internode and form distinct aerating system. Then the cortex is delimited from the stele by an endodermis, which varies in its position in different species of Equisetum. The endodermis is followed by a single layer of parenchymatous pericycle. Then the vascular cylinder, which is at the center, it is siphonostylic in case of Equisetum aerial shoot, where vascular bundles are arranged in a ring around the large central pith. The number of vascular bundles corresponds with the number of ridges. The vascular bundles in case of aerial shoot of Equisetum are conjoint, collateral and end arc. When we cut the transverse section of node, it shows certain different features from that of internode. These features are: number 1, the pith is not hollow at the node, but instead a solid diaphragm called nodal diaphragm is present. Second, vallicular canals are usually not present in the node. Third, the vascular bundles fuse together and form a vascular cylinder around the nodal diaphragm. Fourth, corneal canals are absent in the nodal region. Fifth, leaf and branch traces arise from the vascular cylinder of the node. When we cut the internal structure of the rhizome, it shows certain different features from the aerial shoot, which are as under. Number 1, the epidermis of rhizome is without stomata. Second, the outer cortex of rhizome is less sclerenchymatous. Third, chlorophyll cells are not found in the cortex. Fourth, the vallicular, corneal and central canals are narrow in comparison to those in the aerial shoot. Now let us discuss the internal structure of root. A transverse section of root shows epibelema, cortex and stele. The single-layered epibelema forms the outermost protective covering of the root. Some cells of epibelema elongate to form root hairs. The epibelema is followed by a wide cortex, usually differentiated into an outer sclerenchymatous and an inner parenchymatous region. The latter has intercellular spaces. The sclerenchymatous outer cortex is also known as exodermis. A characteristic feature of the root of Equisetum is the presence of a two-layered endodermis. The cells of the outer endodermal layer are larger and have Casparian bands or strips, but those of the inner endodermis are smaller and without Casparian strips. Pericycle is absent and the lateral roots arise from the inner endodermis. A protostele is present in the center of the root with diarch to tetrarch xylem. A large metaxylem tracheid is present in the center of stele and the protoxylem strand lie around it. The phloem is present in between the protoxylem strands. Equisetum reproduces by vegetative as well as asexual methods. First, we will discuss vegetative reproduction. The underground rhizome of some species of Equisetum. For example, we have Equisetum arvense and Equisetum telemachia. It forms tubers which help in vegetative propagation. These tubers develop due to irregular growth of some branch bud at the nodes of the rhizome. They are oval-shaped in case of Equisetum arvense or pear-shaped in case of Equisetum telomecia. After their detachment from the parent plant, the tubers remains in the soil, and on the return of favorable conditions, they grow into new plant bodies. Production by spores is also called as spore formation. Equisetum is a homospore stereophyte, which means that all the spores produced are alike. The spores develop within sporangia borne on sporangiophores. The sporangiophores are aggregated into a compact cone or strobilus. The position and structure of the strobilus. In most of the species, strobili are borne terminally on vegetative shoots, but in some species, like in case of Equisetum arvense, they are borne on special fertile shoots. The strobilus has a central massive axis called strobilus axis and large number of sporangiophores. The sporangiophores project at right angles in successive alternate whorls from the strobilus axis. Each sporangiophore is a stalked structure with a hexagonal peltate disc at its distal end. On the underside of the sporangiophore disc, 5 to 10 sac-like sporangia are borne near its periphery in a ring. The free round apices of sporangia face the strobilus axis. The development of sporangium, how it develops. The apical growth of the shoot, which is destined to bear strobilus, is slowed down and it assumes a conical shape. This conical apex now functions as strobilus axis. The primordia of sporangiophore are formed on the axis in an acropetal succession in position of leaf primordia. Initially, the primordium is hemispherical, but soon it becomes peltate due to the formation of a constriction near a base. The development of sporangium in case of Equisetum is eosporangiate. That is, a sporangium develops from a group of initials. Sporangia are initiated near the center of the apical dome of the sporangiophore, but later the dome expands laterally, and as such the sporangia are pushed to the margins and finally to the underside of the peltate disc. The first division of the sporangial initial is periclinal, establishing an outer and an inner daughter cell. The inner daughter cell forms sporangious tissue by repeated transverse division in all possible planes, whereas the outer daughter cell divides both anticlinally and periclinally and forms 2 to 7 layered sporangial wall. The innermost layer of the sporangial wall functions as tapetum. The cells of the tapetum are irregular, thick-walled, and nutritive in function. The cells of the outer wall layer develop thin bands of helicoid secondary wall thickening, which presumably help in the dehiscence of the sporangium. As the sporangium matures, all out of the 2 outermost wall layers disintegrate and form a periplasmodial liquid. At this stage, the sporogenous cells separate from each other and function as spore mother cells. Before meiosis, about 30% spore mother cells also disintegrate and form plasmodial fluid. The remaining spore mother cells remain active and float freely in the periplasmodial fluid. Each spore mother cell undergoes a reduction division and forms 4 spores. The spores soon become spherical. The mature sporangia are sac-like structure attached to the underside of the peltate disc of the sporangiophore. The wall of the mature sporangium is only 2-layered. All the spores in a sporangium are alike. As the sporangium matures, the strobilus axis elongates. Consequently, the compactly arranged sporangiophores separate from each other and the sporangia are exposed. As the sporangium dries, the helicoid thickening bands present in the outer wall layer shrink and the sporangium ruptures. The spores of Equisetem are spherical, uninucleate, green, and contain numerous chloroplasts. The spore wall is differentiated into 4 concentric layers: the outermost perispore, the second middle layer, the third exospore, and the innermost endospore. The perispore, also known as epispore, splits into 4 strips or bands. Although these bands are separate from each other, they are attached to a common point on the spore. Initially, these bands are wrapped around the spherical spore, but as the spore dries, the bands are uncoiled. These bands are known as elaters, and they have expanded spoon-like tips. The elaters are hygroscopic and respond to change in humidity. Although their definite function is not known, they probably help in dehiscence of the sporangium by expanding and contracting. The spore of Equisetem germinate shortly after shedding. They are short-lived and remain viable for 1 to 48 hours after shedding. Such a short duration of viability of Equisetem spores is perhaps due to their high rate of respiration. These spores germinate on a suitable substratum. They swell by absorbing water and shed their outer coat. The first division of the spore is asymmetrical, producing a rhizoidal cell and a larger cell. The former develops into the first rhizoid, whereas the latter eventually gives rise to prothallus. The shape and size of prothallus depends on prevailing conditions. If the number of spores germinate within a limited space, the prothalli formed are usually filamentous, while some germinating spores usually form relatively thick and cushion-shaped prothalli. Both antheridia and archegonia develop from the superficial meristematic cells of the prothallus. Archegonia are confined to the basal cushion region of the prothallus, and the antheridia on the lobes of the upright branch. Usually, archegonia develop before antheridia. The development of archegonia first. Any superficial cell from the margin of the prothallus functions as archegonial initial. This cell divides periclinally into an outer primary cover cell and a central cell. The primary cell divides by 2 vertical divisions at right angles to each other, forming 4 primary neck cells. These cells divide further and form 3 to 4 celled high neck of the archegonium. By the time the central cell divides transversely into a primary neck canal cell and a ventral cell. In the meantime, the ventral cell forms 2 unequal cells by transverse division. The larger basal cell forms egg, and the small upper cell forms as a ventral canal cell. The mature archegonium has a small neck and a swollen base. The neck is made up of 3 to 4 tiers of neck cells. Each tier usually has 4 cells. The neck canal has 1 or 2 neck canal cells. The swollen basal part of archegonium has a ventral canal cell and an egg. The neck of archegonium usually projects above the surface of the prothallus. In monoecious species, antheridia develop after archegonia. There are usually 2 types of antheridia in case of Equisetem: projecting antheridia and embedded antheridia. The projecting antheridia develop on the margins of prothallus or on the lobes of the upright branch, whereas the embedded antheridia develop in the massive cushion-shaped part of the prothallus. The superficial antheridial initial divides periclinally into an outer jacket initial and an inner androgonal cell. The jacket initial divides only anticlinally and gives rise to a single layered antheridial jacket. A large triangular jacket cell at the apex of antheridium differentiates into a cover cell or opercular cell. The nucleus of the androcyte progressively changes in shape to become helicoid. Then each androcyte metamorphoses into an antherozoid. The interior part of the antherozoid is spirally coiled and has numerous flagella, whereas the posterior part is somewhat expanded. Before fertilization, the neck canal cells and ventral canal cell disintegrate and form a passage for the entry of antherozoids. The disintegrated mass of these cells contain malic acid, which attracts freely swimming antherozoids towards the neck of the archegonium. Although many antherozoids move towards an archegonium, only one of them swims into it and fuses with the egg. Thus zygote is formed. Usually more than one archegonia are fertilized in a prothylus. For example, in case of Equisetum debile, as many as 15 young embryos have been observed in a single prothylus. The diploid zygote formed by the fusion of male and female gametes is the mother cell of next sporophytic generation. It develops embryo which form a new sporophyte. The embryogeny of Equisetum shows variations in different species. In most of the species, the first division of zygote is transverse, establishing an epibasal and a hypobasal cell. Both of these cells contribute to the formation of embryo. The second division of the zygote is vertical, and as such, a 4-celled embryo is established. All the 4 cells of embryo are of different shape and size. In case of Equisetum arvense, 4-celled embryo divides by second vertical division at right angles to the first one, forming 8 cells arranged in 2 tiers of 4 each. The largest of the 4 epibasal differentiates as apical cell of the shoot, and the remaining 3 cells form leaf initials. The largest of the hypobasal cells functions as root initial, and the remaining 3 cells give rise to fruit. Active divisions in the apical cell of the shoot and root initial results in the establishment of the shoot and root. The embryonic root penetrates the prothylus and enters the soil. The first shoot, called primary stem, bursts through the neck of archegonium and grows upright. It is differentiated into nodes and internodes. Each node of the primary stem has only 3 leaves. The primary stem shows only limited growth. Its apical growth ceases after 10 to 15 internodes have been formed. Then a secondary branch develops at the base of the primary stem from a bud. It has 1 to 5 leaves at each node. The secondary branch also has limited growth and is replaced by another upright branch arranged from its base. This process may be repeated until 3 to 4 shoots appear. And finally, a shoot, instead of growing in usual upright position, grows horizontally and penetrates the ground, forming the underground rhizome. It is perennial and gives rise to new aerial shoot regularly. In the life cycle of Equisetum, we find alternation of generation. Equisetum plant is a sporophyte, which is the dominant phase in its entire life cycle. The sporophyte comprises rhizome, aerial branches, roots, and leaves. It reproduces asexually both vegetatively as well as by means of spores. Equisetum is a homospore stereophyte. It produces spores which develop within sporangia borne on sporangiophores. The sporangiophores are aggregated into a compact coon or strobilus. Each spore germinates on a suitable substratum to form gametophyte, which bear antheridia and archegonia. The antheridia and archegonia respectively form antherozoids and egg, which on fertilization form zygote. The zygote undergoes several divisions to form sporophyte, which bear shoot and root, and thus once again starts its life cycle.